Mitosis is the process of cell division and repair in eukaryotic cells, asexual reproduction.
At the start of metaphase, the mitotic spindle has already formed, and the centrosomes have already migrated to opposite ends of the cell. The replicated chromosomes attach to the spindle fibers at the centromeres, which pull them back and forth until they are completely aligned in the middle of the cell. This, in turn, will help to facilitate an even split of the sister chromatids in the future between daughter cells during anaphase.
In anaphase, the mitotic spindle attached to each chromosome centromere shortens, therefore splitting apart the sister chromatids and reeling them into opposite sides of the cell. The sister chromatids are no longer attached, so now they are not replicated, but they are identical.
While mitosis simply produces two identical daughter cells from any kind of parent cell that can divide, meiosis is specifically the process of creating haploid sex cells, or gametes, for use in sexual reproduction. At the end of prophase 1, the homologous pairs of chromosomes have joined together as a tetrad. This is when a phenomenon called crossing over occurs, where the ends of the internal sister chromatids of the tetrad overlap. Here, genetic material is exchanged or literally swapped between the two homologous pairs, as seen by the mismatched colored tips. This further enhances the variation that exists in meiosis, a kind of shuffling of the genetic deck. Then, in metaphase 1, the homologous pairs attach to the spindle fibers at each centrosome, which, after shifting the pairs back and forth, finally align them randomly in the middle of the cell. Again, this ensures that each new daughter cell receives exactly one homologue from the parent cell.
Another way that meiosis achieves variation is through independent assortment, which occurs in metaphase 1. Independent assortment is the random order that the tetrads line up. When the tetrads are separated in anaphase, the new daughter cell could end up having all maternal chromosomes or all paternal, or a variation of both. The formula for the amount of variation independent assortment can make is 2 to the nth power, n being the amount of homologous pairs. So for humans, it would be 2 to the 23rd, which makes more than 8 million different possible combinations. That is a lot of variation. Next, in anaphase 1 of meiosis, the microtubules that are attached to the homologous pairs contract, pulling the one homologue away from the other towards opposite poles of the cell, while those that are not attached expand, lengthening the entire cell.
Metaphase 2 of meiosis is actually quite similar to metaphase of mitosis, but in metaphase you would not see the mismatched tips that you do see in metaphase 2 because there is no crossing over in mitosis. During metaphase 2, in each daughter cell, the microtubules of the spindle attach to the chromosomes on both sides of each centromere and begin to pull them towards the middle of the cell. At the end of metaphase 2, the chromosomes are completely aligned in the middle, placing them in prime position to be evenly divided among the 4 daughter cells after anaphase 2.
Finally, in anaphase 2, also in both daughter cells, the microtubules separate the sister chromatids, pulling the chromosomes to opposite ends of the cell, and like the other anaphases, the other microtubules extend, elongating the cell.
From here, due to the chromosome's positioning, it is relatively easy for the two parent cells to divide into 4 daughter cells.